Hi, everyone. My name is Joanne Gold, and I'm the Executive Director of the Scleroderma Research Foundation. I'm pleased to be able to introduce our third forum topic for today, the Conquest trial platform. Conquest is a highly innovative clinical trial platform and is designed to rapidly advance promising new treatments for scleroderma. It's a groundbreaking effort, and we're delighted to be able to share some information about it with you today. But before we begin, I have a few quick notes. During this entire session, you can submit questions in the Q&A panel on the left side of your screen. At the end of the presentation, we'll answer as many questions as we have time for live. Then all other questions will be answered via direct message today or by email after the forum has concluded. There'll also be a short survey at the end of the Q&A session, and after a brief break, the next session will automatically begin. Now it's my honor to introduce our two next speakers. First, we'll hear from Dr. Toby Maher, who is a professor of clinical medicine and the director of interstitial lung disease program at Keck School of Medicine at the University of Southern California, Los Angeles. Dr. Maher has spent the last 20 years specializing in the management of all forms of pulmonary fibrosis and orphan interstitial lung diseases. Previously, he led the ILD unit at the Royal Brompton Hospital, London. He remains a professor of respiratory research at Imperial College London, where he is the British Foundation Chair in Respiratory Research. His research interests include biomarker discovery, the lung microbiome and host immune response in the pathogenesis of IPF, and clinical trials in interstitial lung disease. He has been involved in more than 70 trials in fibrotic lung disease, from phase 1B through to phase 4, and including those assessing IPF, scleroderma, rheumatoid arthritis, and inflammatory myositis. Dr. Maher is also an associate editor for the American Journal of Respiratory and Critical Care Medicine and is on the editorial board of the Lancet Respiratory Medicine. He has authored over 350 papers and book chapters on pulmonary fibrosis and has served as an expert to the FDA and European Medicines Agency. Dr. Maher has also been a critical contributor to the formation of Conquest, and he is currently its co-principal investigator. Next, we'll hear from Dr. Dinesh Khanna. Dr. Khanna is the Frederick G.L. Hutewell Professor of Rheumatology at the University of Michigan, Ann Arbor, where he leads the university's scleroderma program. In this role, he directs a multidisciplinary group of caregivers, scientists, and clinical researchers dedicated to advancing knowledge about scleroderma and the related conditions. Dr. Khanna's research interests include clinical trial design and evaluating new treatments for scleroderma. Dr. Khanna is also funded by the NIH to develop new patient-reported outcome measures in patients with scleroderma and related diseases, and is spearheading initiatives to work with the FDA so that those measures can support drug approvals. In addition, he is leading international efforts to develop guidelines for the management of scleroderma and gout. Dr. Khanna is also a trusted advisor to several pharmaceutical companies seeking to understand scleroderma and the issues in drug development for this disease. Dr. Khanna has been a critical contributor to the formation of Conquest, and he is currently its principal investigator. And now, let's get started. Toby, we'll begin with you.
Thanks, Luke, for the introduction. So it's a real pleasure to be here today talking about the Conquest trial. So I'm Toby Maher. I'm a pulmonologist. I'm the director of interstitial lung disease at the University of Southern California in Los Angeles. And I'm one of the co-PIs of the Conquest study. Joining me also for this talk is Dinesh Khanna. I'm sure he will introduce himself, and probably he needs no introduction, but he is director of the Scleroderma Center at the University of Michigan. Again, a lot of what I'm going to say is, I'm sure, pretty well known to those of you listening, but it's always worth going back over it. So scleroderma, as we know, is a systemic autoimmune condition that affects multiple organs, classically the skin, but also many of the internal organs. And importantly, of the internal organs affected, the lung is most commonly so, with about 70% or more of scleroderma patients having interstitial lung disease. And as we know, interstitial lung disease is characterized by inflammation and scarring around the little air sacs in the lung, And that inflammation and scarring ultimately leads to impairment of lung function and the symptoms that patients suffer with. We know that interstitial lung disease is more common in certain groups of scleroderma patients, so particularly individuals with the anti-topoisomerase antibody or the anti-SCL70 antibody. And we know that in some patients it can be a trivial problem, whilst in others it can be a severe, life-threatening and even life-shortening condition. And as many as a third of people with interstitial lung disease in scleroderma will have disease that gets progressively worse over time. And as a pulmonologist, one of the challenges that I encounter is that once the lung is scarred and damaged, that tends to be an irreversible process. So a lot of what we're trying to do when we see patients in our clinic with scleroderma is ideally to prevent the development of fibrosis that's going to cause irreversible problems for our patients. And so just to think about how it affects patients in practice, well, what's interesting about the lung is it has a huge amount of spare capacity. That's why people can climb Mount Everest or run marathons, because we have a lot of our lungs that we don't use day to day. And what that means from a practical sense is that people can have lung disease without realizing it. If the most active you are is walking to the shops or running to catch a bus, then it's unlikely you will notice the earliest stages of lung disease. And so often for patients newly diagnosed with scleroderma, they're not aware that they have any lung problems. As interstitial lung disease becomes more advanced and more severe, then patients will often report breathlessness. Classically, that begins only on exertion. So in the earliest forms of the condition, patients will be aware that they become breathless when they're walking around, or that they struggle to walk and talk with family members. As the disease becomes more severe, then often those patients will need oxygen to maintain their breathing and allow them to undertake activities of daily living. The other symptom that we frequently see is cough. This tends to be dry and non-productive, and often will occur throughout the day and tends to be a very annoying and disturbing symptom for patients. Patients will also often report a sense of fatigue and weakness. That often comes from the fact that with the lungs not working as well as they should, undertaking activities of daily living requires more effort and burns more calories. And as a consequence, they feel exhausted in the way that those of us without lung disease feel after undertaking strenuous exercise. And often that's accompanied by a sense of discomfort. Patients are also more prone to the consequences of infection. So with infection, they'll often have heightened symptoms. How do we diagnose interstitial lung disease in clinical practice? Well, the best way to diagnose interstitial lung disease in patients with scleroderma is to undertake CT scan. With the CT, we can see the structure of the lungs and we can very clearly see the development of interstitial lung disease. The other test that we frequently use in our pulmonary clinics is lung function testing. And with that, we undertake two sorts of measures. We measure the size of the lungs by getting people to blow out hard and fast. That's called the forced vital capacity. And that will tend to shrink, so the values will get smaller as the interstitial lung disease gets worse. And in clinical trials, the forced vital capacity is the best way for us to monitor the effectiveness of treatment. The other measurement we do is something called the DLCO. And essentially what we're trying to measure with that is how well the lungs get from how well the oxygen gets from the lungs into the bloodstream. Again, that tends to be affected by the interstitial lung disease as it progresses, although from a clinical trial point of view, it's not such a good measurement to use. And then how do we treat interstitial lung disease in patients with scleroderma? Well, thankfully, over the last 10 or 15 years, we have seen a number of clinical trials that have had positive readouts. Of the drugs on this screen, 2 of them are approved in the United States as treatments for scleroderma-associated ILD. So that's tocilizumab and nintedanib. But there are other drugs that we can use as well. So often the commonest drugs that we use are immunosuppressant drugs. We use those because, as I said at the beginning, we know that scleroderma is caused by overactivity of the immune system. And so drugs like cyclophosphamide, mycophenolate, and tocilizumab all target that activity of the immune system. And in so doing, we know that they can be effective in reducing or even preventing the progression of lung disease. And then more recently, we've seen the development of antifibrotic drugs. So these drugs were developed for a slightly different lung disease called idiopathic pulmonary fibrosis. What they're designed to do is try and prevent the development of further scar tissue in the lung. And they've proved very effective at doing that in idiopathic pulmonary fibrosis. More recently, we've seen clinical trials in patients with scleroderma. And again, these drugs have been shown to slow down the rate at which fibrosis accumulates. So for patients where immunomodulatory therapy has been ineffective, we will often consider using antifibrotic drugs now. And then, of course, for patients who really haven't responded to treatment or who have very severe disease, then we have the option of transplant. And more recently, we've been using autologous hematopoietic stem cell transplant as a way for us to be able to give very high doses of immunosuppression, or for a longer period of time, we've been using lung transplant. But both of these transplant-based approaches have challenges. They carry considerable risks associated with them, including risks of death. And also they require that patients are on long-term immunosuppressants. So if we can find ways to avoid transplant, then that's much better for patients. And so how do we develop new treatments? Well, we do that through clinical trials. Many of you, I'm sure, have been involved with clinical trials. Whilst in principle, clinical trials are a great idea, the practical reality is that they are very complex and expensive and difficult to deliver. Your average phase 2 trial, so a trial with perhaps 100 to 200 patients in it, will cost anywhere between $30 to $100 million. It will take often 12 months to complete the necessary paperwork to be able to run the trial, and recruitment of that trial will then often take 1.5 to 2 years. And as a consequence, these trials will run from the first idea, the first concept, through to the results will often be a period of 4 or more years, making the drug development process very slow. And there's a lot of things within trial delivery that could be streamlined. So one of the difficulties when delivering clinical trials is that each center involved, so each individual hospital, has to sign a contract with the sponsor who runs the trial. And that contracting process often takes very many months. We then have to get regulatory and ethical approval through the IRB. Again, that takes time and effort and often requires many people to do it. There's then the training of sites. So at each site, there will be study nurses and coordinators. There will be the trial investigators. Each of them need training. And what tends to happen at the moment is that with each individual trial, that process starts afresh each time. So the contracting, the IRB approvals, the training of staff, and the running and delivery of studies. And so the concept with platform trials, as you will hear from Dinesh, is that we streamline this process by allowing a single trial protocol to deliver studies using multiple drugs. And that cuts out a lot of the time-consuming steps that go into delivering trials at present. And for a rare disease like scleroderma, where there are relatively few patients with this condition, this streamlining is going to be really important to improve the efficiency of trial design and delivery. So I'm going to stop here and I will hand over to Dinesh, who's going to take you through a description of the Conquest trial and explain to you why this is going to represent a real step change in trial delivery for patients with scleroderma-associated ILD.
Hello, everybody, and good day. My name is Dinesh Khanna. I'm a rheumatologist and the principal investigator for the Conquest protocol. You just heard Professor Toby Maher talk about the unmet need for interstitial lung disease or lung fibrosis with scleroderma. My job today is to really present you what we are doing in collaboration with the Scleroderma Research Foundation, the Conquest protocol. Conquest is a clinical trial. It's a multi-center, global, double-blind clinical trial in patients who have scleroderma-related interstitial lung disease or lung fibrosis. It's a very exciting time because Scleroderma Research Foundation is launching the first ever scleroderma clinical trial platform that I will define a little bit more in detail in the next slide. Now, it's a phase 2B trial, and for our patient partners, you may be wondering, what does a phase 2 trial means? There are 3 large clinical trials, 3 large phases. Phase 1 is to look at the safety of a drug in human beings, whether it is healthy volunteers or it's people with scleroderma-associated lung fibrosis. Phase 2 is an intermediary stage where we look at the efficacy and safety of a drug in patient population of interest. It would be scleroderma lung fibrosis here. And phase 3 is really the registration trial with the Food and Drug Administration to get it to the market and to the patient with scleroderma and lung fibrosis. It has a lot of efficiencies built in. We are incorporating a variety of pharmaceutical industry partners to have access to novel agents and financial support. I think one thing I would repeatedly highlight here is that it's picked the winner platform and really has been designed to maximize patient welfare. So here is a study design. It's a bit complicated, and I'll take some time to walk you through this. Our inclusion criteria needs people who have scleroderma-related lung fibrosis, as highlighted by Professor Maher. Patients who have scleroderma lung fibrosis with certain inclusion and exclusion criteria, so that we recruit the right people for the right drug, and this would be publicly available soon on clinicaltrials.gov. People with scleroderma-ILD come into the trial, they meet the inclusion and exclusion criteria, and then they are randomized to either one of the 3 hypothetical therapies. Therapy A from company Y, therapy B from company Z, and therapy C from company D. All these patients, you know whether you're being randomized to therapy A, therapy B, or therapy C, but you and I will be blinded whether you get the active drug or the placebo. I didn't talk about these stratification factors. These are factors that may influence the primary outcome measure, which is a forced vital capacity, your lung function test, and the outcomes may be different whether you're taking CellCept or mycophenolate or whether you have limited or diffuse scleroderma. So going back to our therapies, we know that you are randomized to therapy A. So where is the efficiency? What's in for the patient? There is 3 to 1 chance that you will get active therapy compared to placebo. So let's assume we want to recruit 100 patients with active drug and 100 patients in placebo. In a traditional clinical trial that you may participate, there's 1 to 1 chance that you may get the active drug or you may get the placebo. Here, the placebo is shared among the 3 different therapies. So therapy A will contribute 33 patients. Therapy B will contribute 33 patients, and therapy C will contribute 33 or 34 patients. Whereas in active therapy A, 100 patients will be randomized to therapy A, 100 patients to therapy B, and 100 patients to therapy C. So this is where the efficiency, the patient welfare is kept in mind while designing this trial. It's a 48-week trial. Let's assume all the 3 therapies start at the same time. Therapy A, therapy B, and therapy C ends the trial at 48 weeks. Once the trial is finished, therapy D, E, and F respectively can come and dock into the areas where therapy A, B, and C were already there. And there are different outcomes. Let's assume therapy A was shown to be effective and safe. The pharmaceutical company can then launch a phase 3 trial, a registration trial with the Food and Drug Administration to get the drug to the market. Therapy B was found to be ineffective or the safety profile was not favorable compared to benefit. Therapy B drug development can end there. So it's highly effective, highly dedicated trial for people who have scleroderma-related lung fibrosis. It's a very ambitious protocol. We are launching 130 sites worldwide in United States, in Japan, in Europe, and other countries such as Latin America. We really have incorporated, got a buy-in from the international community, both from rheumatologists and pulmonologists and other researchers. So what are the benefits for all the stakeholders? There's a reduced time to complete. When you are able to launch a platform, there is reduced time, efficient startup, time and effort required to set up new regimen, contracting. All sites are open once and they remain open perpetually. High quality execution. We have been able to incorporate high quality sites around the globe with investigators, uniformity of data collection, recruitment and retention strategies, and other shared services. The third bullet point is very important to our patient partners. Like I stated before, I would like to state it again. Reduced number of patients on placebo because placebos are shared, really appealing for any patient participating in a clinical trial, and also reduced cost because fewer total patients are required for each group. Better disease learning. The placebo group, which is 100 patients, will be shared by the community, owned by the community and Scleroderma Research Foundation, then can be utilized for drug development and enrichment for future clinical trials. There's an independent therapeutic selection committee. You heard Professor Peter Merkel today. He's part of the independent therapeutic selection committee, and they have a very important role. Their role is if there are 10 different drugs and 10 different pharmaceutical companies who have interest in this platform, they come with their data, and this committee looks at the biological plausibility in people with scleroderma-related lung fibrosis. Their task is to review the clinical pharmacology, to look at the route of administration, the safety, the preclinical data in humans, in animals and others, before they make a decision whether this drug should be incorporated as part of future platform therapies. And here is the Conquest study structure. It has taken us really 3 to 4 years to put this together. There's a Conquest platform and Scleroderma Research Foundation leadership that includes me as a principal investigator, Dr. Toby Maher as a co-principal investigator, Dr. Luke Evnan, and Dr. Wade Benton, amongst others. We have been able to galvanize the international community, and they are part of the steering committee, providing critical insight and input during the trial design, and also includes an advisory committee of patient partners and Scleroderma Research Foundation members. I think one of the critical aspects that is overlooked and not talked enough with the patients is something called data monitoring committee. They probably have the most important role to safeguard the safety of the patients. They meet on regular basis. They can look at the unblinded data for both efficacy and safety, and they can stop or halt the study or refine the study if needed, if they believe that the risks are outweighing the benefits. We have highly experienced international statisticians, a pulmonologist and a rheumatologist who will be part of the data monitoring committee. There's a Conquest Scleroderma Research Foundation team. There are pharmaceutical partners. There's something called a CRO, which is a contract research organization, MedPace, that is responsible for execution of the study at each site, data collection, data management, monitoring of the study, and the data collection. We talked a bit about the therapeutic selection committee, and finally, we have task force, experts and researchers around the globe that will be looking at biomarker discovery, novel outcome measures, how can we expedite the drug development and decrease the duration of the trial for a go, no-go decision. This has been a long journey, a journey that has been filled with trials and tribulations. This proposal or this concept started with Dr. Evnan, myself, and others about 5 years ago, but we really put this in action in late 2021 by putting the platform and the protocol together. We went to the Food and Drug Administration and received enthusiastic feedback from them. And starting later 2022, we had the study onboarding, kickoff study implementation, and had recruited MedPace to join our team. We are now in the mid of 2023, where I'm excited to say we have 2 pharmaceutical partners who have agreed to be part of this initiative. So it's a very exciting time for all the hard work and effort that we have done here. We will be putting IND, which is a new drug submission, to the FDA for each of those therapeutic agents, with the planned recruitment of the first patient in November of this year, November of 2023. Very ambitious timelines, like I said before, almost 400 patients around the globe at 130 sites will be recruited in a matter of 15 to 18 months, with a go, no-go decision for a phase 3 program in April of 2026. So here's my final slide, why you should participate in Conquest. Again, highly effective, highly efficient trial design with a single platform for a go, no-go decision for a phase 3 trial. You are being part of the drug discovery, biomarker validation, outcome markers development. If you are a patient, it's highly efficient because you're more likely to get the active therapy compared to placebo. And like I stated, improving the design and timeliness of this protocol. None of this would be possible without the support of the Scleroderma Research and Foundation, and especially Luke Evidence. So thank you very much for listening to my talk.
This is Dr. Peter Merkel. I'm back again with Dr. Khanna and Ma. And thank you both for an excellent presentation about this, I think, incredibly exciting platform for research. This is the most transformative change in scleroderma research in a very long time, and really has the potential to help us to really find research to help patients with lung disease and perhaps beyond. And actually, we have a number of questions that have come in, and I think we're going to try to go for relatively short questions, relatively short answers, so we can get as many answers as possible done. For those of you who are submitting questions, you can submit answers as well. That don't get live, the Scleroderma Research Foundation will try to provide, and will certainly provide, some answers that they will post after this session. So let's get into it. One of the things that comes up is, what if you have scleroderma lung disease for greater than 5 years? What are the options for you? And that sort of segues into, do all studies have these criteria? There are other options for research. Why don't we start with Dr. Khanna about the 5-year. Why is it 5 year, briefly, and what other options? And then we'll turn it back to Mo. And I may answer some things too.
Yeah, thank you, Dr. Merkel. We didn't talk in detail about inclusion-exclusion criteria because they will be publicly available on clinicaltrials.gov. But the recruitment for this protocol is limited to 5 years because we want an enrichment group, a group that is likely to progress over time and able to differentiate drug from placebo. Remember, this is a phase 2 protocol, and I highlighted what a phase 2 protocol is in my talk. And we want a quick go, no go decision to move forward in this trial to go to a phase 3 registration trial. There are many options that we have that, as you and Dr. Maher are aware of. We tend to use immunosuppressive therapies from mycophenolate mofetil to rituximab to tocilizumab, or antifibrotic therapies such as nintedanib. Both tocilizumab and nintedanib are FDA approved for management of scleroderma-related interstitial lung disease.
So,
Dr. Maher, your thoughts?
Yeah, specifically about the inclusion of people into trials, one way or the other, the 5-year approach. Dr. Ma?
Yeah, and I think, one of the challenges running clinical trials is that we do need to have inclusion and exclusion criteria. And the reasons for that are that we, at the end of the day, we need to show that the drug works by showing a difference between the placebo group and the active treatment group. And we use the criteria to try and select patients whose disease we anticipate will get worse more quickly, because that makes it possible to show a treatment effect more easily. And our general sense from what we know about scleroderma ILD is that for patients who've had the disease for 5 years or more, the ILD tends to be less of a problem. It's less progressive. It gets worse much more slowly, which obviously is good for that individual, but from a clinical trials point of view, then makes it very hard to show a treatment effect. And so
Yeah.
we have the rules for that reason, but I do understand why for patients it's very frustrating that sometimes they can't get enrolled in trials.
Yeah, we seem to lost Dr. Khanna. I hope he reappears. So I'll just take on the piece about what other research. So first of all, I understand it is frustrating. Some patients, it's fantastic that patients want to participate in research. And I think patients with scleroderma are particularly motivated knowing that we have a long way to go to get this disease where we want to be for our patients. But I would remind everybody that these are experiments, as I said in my talk, so let's not have what we call therapeutic misconception. These are experiments. And so, we want to make sure the experiment is properly done so it's safe. If you are, so there's a couple of reasons you may not be eligible because you have another problem, other medical issues. We're pretty tight about making sure we do it safely with new drugs for new diseases. Now, there are other trials out there for interstitial lung disease. And I think, as I mentioned, you can look through on clinicaltrials.gov. You may have to learn to put a few filters in, scleroderma plus lung disease. Additionally, I know there are people who don't live near a research center. Now, there's sometimes other ways to participate in research. Sometimes there's online systems, etc. And you'd be surprised what comes along. But we understand that people want to participate, and we really appreciate that. So in some cases, as I say to patients, I don't want you to be eligible for this study because it means you have active lung disease. But if there are these options. So there's lots of ways to participate. So the couple of other, this is a good question. Are there treatments for ILD? Now I want to focus on the trials, but this comes up as it relates to Conquest, perhaps, Dr. Ma. Are there treatments for ILD that interfere with the simultaneous treatment of scleroderma? And I'm going to interpret that question to mean is, well, if we're treating the lung disease, are we going to mess up treatment for other parts of scleroderma? Or is it the other way around? What do you think?
I think the ideal hope and goal with these treatments is that we'll not only see benefits for the lung, but that we will see benefits for other aspects of scleroderma. And we recognize that there is a relationship between the mechanisms that underlie the fibrosis and thickening of the skin that are very similar to the mechanisms that underlie the fibrosis that we see in the lung. So in the ideal situation, the same treatment will provide benefit for lungs and skin. Clearly, part of the reason we do the trials is to assess the safety of the drugs. And one of the components in assessing the safety is obviously to make sure it doesn't upset other organs in the body, but also to make sure it doesn't interact with other commonly used treatments for scleroderma. And certainly it's always a consideration as to how well different drugs interact. For instance, with the nintedanib trials, we actually saw a seemingly positive interaction with mycophenolate. But obviously, in doing the study, one of the things we wanted to make sure was that there wasn't a negative interaction, and that's always a very important consideration. And it does come back to some of the inclusion-exclusion criteria that if we think there will be a negative interaction between drugs, for instance, we would exclude people from being on those drugs when being in the study.
That's great. I'm going to switch now. We have a series of questions about Conquest, and people are already excited. So, Dr. Khanna, I'm going to rope a few questions in together. Some of our, I believe that patients have posted questions. One of them lives in Nigeria. One of them lives in Ontario. And when can they participate? And are there plans to expand this to Europe? And let's do some brief answers here.
So absolutely, I told you 130 centers. So far, we are in Europe, North America. We are trying to activate Canada. So somebody in Ontario should be able to participate. There is no plan to go to Africa, but this is a first step. I want to highlight this is a perpetual platform, and the goal is we are starting with 130 centers, but the goal is to make it truly an international endeavor and go to every country, continent that we can go to be able to achieve it.
And There's another question I'll take, which is, has this platform been used for other drug studies in other diseases or rare diseases? So the classic is in oncology, a study called I-SPY for breast cancer. And I think we certainly know that this was somewhat based on that approach to this platform study. Our oncology colleagues have done a lot of studies through that platform, and there are other disease groups that have done this as well. This may be one of the more focused ones on a relatively rare disease, but I will tell you, I know this sounds funny, but sometimes they do it in subsets of breast cancer or subsets of another cancer, where it's almost its own rare disease within it. So the platform works on a relatively small and medium level, and it should work for scleroderma as well. I don't know if there are any other comments, but I think we're borrowing best techniques from other medical professionals to make this the best possible platform study.
And I'm just to jump in very quickly, I think with I-SPY, which is the breast cancer one, they have seen a successful approval of drugs directly from that platform. So it's been a very successful approach, which obviously we're hoping to replicate.
Dr. Khanna, how big can this get? How many different arms could you have? And then I'll say, are there plans to go outside of lung disease and scleroderma?
Thank you, Dr. Merkel. So right now we can accept up to 3 arms because this is a rare disease, like one of our patient partners stated, and we can go up to 3 arms. We are launching with 2 pharmaceutical agents, 2 therapeutic agents, and there'll be a press release next week on which are the companies and which are the drug. Dr. Maher and I, along with you, are very excited about it. There are goals out here to go beyond scleroderma-ILD. I think we in Scleroderma Research Foundation feel this is a first step. Like you and Dr. Maher highlighted, what about people with very early scleroderma, where we are trying to have a disease-modifying effect, not just in the skin, but also in the lungs, quality of life, hand function, and other aspects? Could these go into pulmonary hypertension? Could this go into Raynaud's and digital ulcers? So the options are unlimited, but the key part is to have an agreeable master protocol with similar inclusion and exclusion criteria.
Very good. Here's an interesting one, and I take a stab, but I'll give Dr. Ma a curveball here. How might AI, artificial intelligence, help in research advances in this disease?
That's a very broad question. I think there's a number of ways. One way that AI is being used at the moment is to try and improve our use of lung imaging. So at the moment, when we measure the effect of drug, we're tending to use forced vital capacity, which is the lung function test where people blow hard and fast into the machine. What we think in the future we might better be able to do is use changes in CT scan appearances to tell us whether the drug has modified the effect of the interstitial lung disease. And I think the application of AI there is very exciting. I think other ways we can use it in the future are probably to better identify patients who might benefit from specific trials. And also perhaps to identify patients out there in the wider community so that we can alert them, sort of find them and go to them with the proposition of a trial, rather than having to wait for patients to go to the right center to know about studies. So I think there's lots of ways it can be used.
Those are great answers. I think of AI. There's a tendency to think it's a black box. It's one thing. It's really, think of it as multiple different new mathematical algorithms, sort of supercomputing. It's really a broad range. It could also help identify new targets for drugs. That is almost certainly a way that we're going to use it based on genetic data or clinical data. And as you said, to find the right study for the right patient. So I think there's all sorts of ways that AI and its related methods are going to help us in science. We're not afraid of it. We want to harness it and use it for the good of the science. And we've already been doing that in many fields, including scleroderma. Dr. Khanna, someone said, I'm not sure I understand this Conquest. Hasn't the Scleroderma Research Foundation been funding research for years? The answer is a resounding yes. Can you expand on that?
Oh, absolutely. Scleroderma Research Foundation has been the premier nonprofit foundation that has been funding research. They have really funded high quality, high impact research that has led to knowing the causes of scleroderma and discoveries that have happened over the last decades, a few decades. I don't know how many decades, but it has been funding quite a bit of research. They also funded the first national registry called Conquer. So there's a Conquer and then there's Conquest. And that has almost 17 centers, if I'm not mistaken, around the US. And many of our patient partners may be participating, including you and I, that are part of this proposal. So yeah, we are very, very excited, very honored to be part, and working with Scleroderma Research Foundation to make Conquest a reality.
So we only have 2 minutes left. Let's do some quick wrap up. And again, I think we've gotten to many of the questions. I can ask each of you, Conquest is very exciting. Where do you see it in the next couple of years? And what's it's going to bring to us? Maybe just sum up what we think is going to happen again. I'll start with you, Dr. Ma.
So I think as Dinesh and was said in the talks, that we need to fast forward research for scleroderma so that we can get drugs approved much more quickly. And I really think that this platform trial is a very important way of doing that. I guess in the next 2 years, we want to see rapid and effective recruitment for the study so that we have clear timelines on when these first 2 drugs are going to read out. And hopefully also we'll be thinking about bringing more drugs into the platform by that time.
Dr. Khanna.
I would agree to that. We want rapid activation of centers. We want rapid recruitment. And like Dr. Maher said, we really want a very quick go, no-go decision, because if the drug is not working, it should not proceed forward. We should be able to bring other drugs to this platform and have the opportunity. And you, Dr. Merkel, as one of the members of the therapeutic selection committee, has a very important role. And we are hoping that in 2 years, you will be flooded with 5 to 10 different drugs, so you can choose the right drug for the right patient in this platform trial.
I will say I was just at the European Rheumatology meeting where you were as well, and talking to colleagues in industry and around, and they're already very interested in doing this. I will end in this and say that there is more research being done in scleroderma now than at any time in history. We are optimistic about our ability to get answers to help patients, and whether it be this trial or another study that you're part of. We appreciate everyone who participates in our research, and those of you who don't but are supportive of the programs that are happening. And I think informed patients who know what's going on are the best patients to work with. And I think as a physician, as investigators, we look forward to working with you. We thank the Scleroderma Research Foundation, which is just doing a phenomenal job of helping to support the field. I think we're done with this session. We'll try to get to as many answers offline as we can.
Thank you, everybody.
Thank you.